Hello, I am Emmanuel Orte Blanco, and today I will tell you about a green synthesis work of polypyrrole using antioxidants from hibiscus leaves and orange leaves as doping oxidation agents. Our goal was to obtain polypyrrole at the nano level and see how the type and amount of extract change its optical and structural prop- properties using diffuse reflectance and Raman spectroscopy. We also explored whether iron particles and appeared and how detectable they would be with these techniques. As we conduct the experiment, we first crushed different amounts of hibiscus and orange leaves, dividing them into samples of 5 grams, 10 grams, 15 grams, and 20 grams, which we boiled for 1 hour and then allowed them to rest. For each sample, we used an oxidation method, and after applying this method, we used a centrifuge at 4000 RPM for 2 hours to obtain iron particles. We transferred these containers to the oven to let them crystallize and collect them. After taking them out of the oven, we applied a certain amount of polypyrrole to each sample. We obtained 3 samples of 5 grams of hibiscus and orange, adding zero point 0.1, zero point 0.12, and zero point 0.14 of polypyrrole. 3 samples of 10 grams of hibiscus and orange, adding zero point 0.1, zero point 0.12, and zero point 0.14 of polypyrrole. 3 samples of 15 grams of hibiscus and orange, adding zero point 0.1, zero point 0.12, and zero point 0.14 of polypyrrole, and 3 samples of 20 grams of hibiscus and orange, adding zero point 0.1, zero point 0.12, and zero point 0.14 of polypyrrole. Then, we, with the help of a mixer, we mixed each one for 2 hours at 2, 2015 RPM. Finally, each sample went through a cleaning process and was placed in the oven for 8 hours to ultimately collect each sample and take it for analysis. Talking about the results first, and we diffuse reflectance. The reflectance, which I call R, is how much the sample reflects light. If R decreases, it means that the sample absorbs more. Second, Raman is like a fingerprint of the vibrations of the molecules. It tells us what bonds are present and how the structure of polypyrrole change. Let's look at this reflectance graph. Um, Reflectance graph. Here I am comparing series with 5 grams, 15 grams, and 20 grams of extract. As the amount of extract increases, the reflection are in the visibles goes down, which means the sample absorbs more light from the same data. I build the dot plot, which we use to estimate the optical band gap, the energy the material needs to start conducting. What happens to the optical band gap? Well, it decreases as we add more extract. Raman is an optical technique where I illuminate the sample with a laser and analyze the returning light. A very small portion returns with a slightly different energy because it interacted with the material's vibrations. That difference is graphed as a Raman shift in centimeters minus 1. Each peak is in practice the fingerprint of a bond or vibrational mode. In polypyrrole, 4K areas typically appear 9, 930 to 1000 centimeters minus 1. This is the modes of the ring. Another rank is 1014 to 1100 centimeters minus 1. Another range is 1330 to 1318 centimeters minus 1. And another range is 1580 to 1600 centimeters minus 1. In my examples, as the plant extract increases, the relative intensity of the band 1330 to 1318 centimeters minus 1 grows compared to that of 1580 to 1600 centimeters minus 1, and the peaks within a bit. This indicates more doping, more charge carriers, and greater disordered amorphicity, amorphization typical of doped nanopolymers. Sometimes the peak shift a few centimeters minus 1, indicating that the conterons from the extract interact with the polypyrrole chain. So, as a conclusion, we can say that with the hibiscus leaf, it was not possible to obtain iron particles in any concentration. Why? Well, I think it it well hibiscus leaf it can be used to to do that. Another conclusion is the orange leaf extract works effectively as a reducing or stabilizing agent in green synthesis. And my last conclusion is that green synthesis using orange leaf extract allowed for the production and stabilization of iron nanoparticles. Well, thank you so much for your attention and thanks.